Hi everyone, this is our Excellent Med and I'm an aspiring medical student sharing tips on the pre-med journey. My goal is to guide you along the way to higher education and help you succeed in your path to becoming a medical professional. Welcome to my first video in the MCAT series focusing on Foundational Concept 1, Biomolecules. So today we're going to be diving into the topic of amino acids, specifically their structure. I've written on the side here, what are amino acids? And as seen on the slide, they truly are essential to your success on the MCAT, whether answering the simpler which amino acid questions or seeing common amino acids within broader contexts in passages. There are 20 amino acids to memorize, which are listed on the slides here, including their names, side chains, and structure, and 1-letter and 3-letter codes. Before we get into the nitty-gritty, let's take a step back and see where amino acids fit into the broader picture of biochemistry and human metabolism. A great way to understand this is by looking at crucial proteins in our bodies. There's hemoglobin, there's P53, but today let's look at insulin. So what is insulin? Insulin is a hormone produced by the pancreas and it plays a key role in regulating blood sugar levels. Think of insulin as a key that unlocks cells so they can absorb glucose from the bloodstream, which is essential for energy production and overall metabolic balance. Just like a key unlocks a door, insulin binds to receptors on cell surfaces to facilitate the entry of glucose into cells. So where do amino acids come into play? Amino acids are the building blocks of insulin and many other proteins. Without them, this vital protein and its crucial function could not exist. To continue with the analogy, if we consider insulin as a key, then amino acids are the individual components that make up the key. There are 20 different amino acids that can combine in various ways to create a vast array of proteins, including insulin. So now that we have a sense of where amino acids fit into the bigger picture, let's zoom in and explore their actual structure. So first, every amino acid consists of an amino group, a carboxylic acid group, and a central carbon atom known as the alpha carbon. The amino group NH2 and the carboxylic acid group COOH are bound to this alpha carbon, and then there's a hydrogen atom and a unique side chain, also known as the R group, attached to this alpha carbon as well. So in total, there are 4 different attachments on the alpha carbon. The name amino acid comes from the 2 groups, amino for the amino group and acid for the carboxylic acid group. Each amino acid has this core structure, but the side chains, R groups, varies between all the different amino acids, giving each one unique properties. Most importantly, the alpha carbon is a chiral carbon. This means it has 4 different groups attached to it, making it capable of optical activity. So if you shine a polarized light through an amino acid, the chiral carbon will rotate light because it has a unique arrangement. However, there's one exception, which is glycine. Glycine is a unique amino acid because its side chain is just a hydrogen atom, which means it does not create a chiral center and it's not optically active. So there are a few ways that we can represent amino acid structures, and one of them is using Fisher projections. So Fisher projections are really helpful for visualizing the arrangement of the 4 unique groups around a chiral carbon, and we see them often on the MCAT. So in a Fisher projection, the amino group is placed to the left or right, depending on whether it's in the L or the D configuration respectively. So the L amino acid configuration has the amino group on the left side, while the D amino acid configuration has it on the right side. And that's a little trick to help you during the exam. So these L and D configurations are called enantiomers, and they are mirror images that cannot be superimposed on one another. And the classic example that's given in class is much like your left and right hands. They are mirror images, but if you put them on top of each other, they cannot be superimposed on one another. So to remember which is which, you can think of L as for left and D as for right, since the D comes from dextro, meaning right in Latin. It's really crucial to note that the L form of the amino acids is the only one found in the human body. So to wrap up, we explored how amino acids fit into larger metabolic processes using insulin as an example. We discussed the basic structure of an amino acid, the role of the alpha carbon as a chiral center with the exception of glycine, and the Fisher projections for distinguishing between L and D configurations. So that's a wrap on amino acids and their structure. If you have any questions or need any further assistance, please do not hesitate to reach out via email at rexcellentmed@gmail.com or leave a comment below. I'm here to help you on this journey. Thank you for taking your time to watch this video. Bye-bye.